Hello and welcome to our lesson on the cell cycle and what happens when it malfunctions. I am Nova Ruiz.
I am Irish Anselmo.
I am Bea Christine Lobusta.
Together, we'll explore the stages of the cell cycle, the process that allows cells to grow, copy their DNA, and divide. But sometimes, things don't go as planned. Malfunctions can lead to issues like uncontrolled cell growth and diseases such as cancer. In this lesson, we'll dive into these stages and examine the consequences when the cycle doesn't function properly. So let's get started.
What is the cell cycle? The cell cycle is the process that helps cells grow, copy their DNA, and divide into new cells, essential for growth, repair, and reproduction in living organisms. In this lesson, we'll explore the two main stages of the cell cycle and understand why it is so important for life. Why do cells need the cell cycle? First, growth helps organisms grow larger. Cells multiply so organisms can grow bigger and stronger. Second, repair replaces damaged or dead cells. When you get hurt or cells wear out, the cell cycle replaces damaged or old cells with new ones. Third, reproduction creates new cells for reproduction. In some organisms like bacteria, the cell cycle allows them to reproduce and create new organisms. Now that we know why it is important, let's dive into the steps of the cell cycle. Stage 1. Interphase. The cell's preparation phase. The first and longest stage of the cell cycle is interphase. During this time, the cell prepares itself for division. Interphase is divided into three parts: Gap 1, synthesis, and Gap 2 phases. Gap 1 phase, growth and preparation. Key points: cell carries out normal functions, increases in size, prepares for DNA synthesis. In the Gap 1 phase, the cell focuses on growth. It performs its usual metabolic functions, like absorbing nutrients, and it grows larger size, preparing for the next stage, DNA synthesis. Synthesis phase, DNA replication. Key points: DNA synthesis occurs. Genetic material is replicated. Next is the synthesis phase, where the cell copies its DNA. This is a critical step as the cell must replicate its genetic material to pass on an identical set of chromosomes to its daughter cells. Gap 2 phase, preparation for division. Key points: cell continues growing, RNA and protein synthesis occur, final checkpoint before division. The final part of interphase is the Gap 2 phase. Here, the cell continues to grow and produces RNA and proteins needed for division. It also checks for any errors in the replicated DNA.
M phase. The final phase in the cell cycle. During the M phase, cell cycle occurs into two processes. The first one is the mitosis, when nucleus divides, and the second one is the cytokinesis, when cytoplasm divides. Mitosis has four phases. The first one is the prophase. DNA condenses into visible chromosomes. The DNA condenses into visible structures called chromosomes, and spindle fibers start to form. The second is the metaphase. Chromosome align in the center of the cell. The chromosome line up in the middle of the cell, ready to split. The third one is the anaphase. Chromosomes separate to opposite ends. The chromosomes are pulled apart to opposite ends of the cell, ensuring each side gets an identical set of DNA. And the last one is the telophase. Two new nuclei are formed. Two new nuclei are from around the separate chromosomes, one for each future cell. cytokinesis or the splitting the cell. The final stage is cytokinesis, where the rest of the cell divides into two. In animal cell pinches into two. The cell pinches in the middle, forming two new cells. In plant cells, a cell plate forms a new wall. A new wall called a cell plate forms in the middle, separating the two cells. After cytokinesis, the cell cycle is complete and now have two identical daughter cells.
Malfunctions during the cell cycle. The cell cycle is a carefully regulated process that ensures the proper division and functioning of cells. Each phase has checkpoints to monitor cellular activities. However, when these checkpoints fail, it can lead to serious consequences, including diseases like cancer or genetic disorders. In this lesson, we will explore some of these malfunctions. Normal cell division versus malfunctioning cell division. Normal cell division is highly organized and ensures that each daughter cell receives the correct genetic material. Checkpoints at various phases of the cycle verify if the cell is healthy, DNA is undamaged, and division is occurring properly. When these checkpoints fail, it can result in abnormal cells that harm the body. Malfunction example: cancer. It is uncontrolled cell division, formation of tumors. Cancer is one of the most well-known consequences of the cell cycle malfunctions. It occurs when mutations in DNA disrupt the regulation of the cell cycle, leading to uncontrolled and abnormal cell division. Over time, this creates a mass of cells called the tumor. The third one is the benign versus malignant tumors. There are two types of tumors: benign and malignant. Benign tumors remain localized and can often be removed. Malignant tumors, however, metastasize, spreading through the bloodstream or lymphatic system and forming secondary tumors in other parts of the body. This is what makes malignant tumors particularly dangerous. Causes of cancer: DNA mutations, lifestyle factors, smoking, high-fat diet, genetic predisposition, environmental exposures, radiation, carcinogens, viral infections, HPV causing cervical cancer. Cancer can be triggered by various factors. DNA mutations are the primary cause, which may arise from lifestyle habits such as smoking or poor diet, or exposure to harmful chemicals and radiations. Some viruses like HPV are also linked to cancers such as cervical cancer. Additionally, genetics and family history may increase the risk. Why are tumors dangerous? Cancer cells lack specialized functions, compete with healthy cells for nutrients and blood, can impair organ function. Cancer cells are harmful because they do not perform the specialized functions of normal cells. Instead, they divert nutrients and blood supply away from healthy cells, impairing the body's ability to function. For example, in the brain, growing tumors can put pressure on other regions, disrupting essential processes. Cancer treatment: chemotherapy targets rapidly dividing cells. Surgery removes cancerous tissues. Radiation therapy shrinks tumors with X-rays. Treating cancer often requires a combination of methods. Chemotherapy uses drugs to kill actively dividing cells. Surgery removes cancerous tissues, while radiation therapy uses high-energy rays to shrink tumors. Early detection is critical to improving the chances of successful treatment. The second example of malfunction is genetic disorders caused by cell cycle malfunctions, errors during dividing of sex cells, meiosis, errors during dividing of other cells, mitosis, exposure to substances that can cause birth defects, teratogens. When errors occur during cell division, they can cause abnormalities in the number of structure of chromosomes. These abnormalities can lead to genetic disorders, each with distinct physical and developmental manifestations. So this is the human karyotype. As you can see, the male is in the left side and the female is on the right side. And now we're going to discuss the examples of chromosomal abnormalities. Each chromosomal abnormality leads to unique challenges. The first one is the Down syndrome, trisomy 21, 3 copies of chromosome 21. Down syndrome results from an extra chromosome 21, leading to developmental delays and distinct facial features. So these are the examples of Down syndrome. The second one is the Turner syndrome, 45X0, missing one X chromosome in females. Turner syndrome affecting females involves a missing X chromosome, causing short stature and other physical changes. These are the examples of Turner syndrome. The third one is the Klinefelter syndrome, 45XXY, extra X chromosome in males. Klinefelter syndrome, seen in males with an extra X chromosome, often leads to infertility and developmental differences. So this is the example of the Klinefelter syndrome. The fifth one is the Patau syndrome, trisomy 13, extra chromosome 13, and the Edwards syndrome, trisomy 18, extra chromosome 18. Patau and Edwards syndromes caused by extra chromosomes lead to severe physical and developmental defects, often with low survival rates. And these are the examples of the syndrome.
Structural chromosomal abnormalities: deletion, duplication, translocation, and inversion. Structural abnormalities in chromosomes can also lead to disorders. For example, a deletion removes a portion of the chromosome, as seen in Cri-du-Chat syndrome, where affected children have intellectual disabilities and a characteristic high-pitched cry resembling a cat's meow. Malfunctions in the cell cycle can lead to life-threatening diseases and genetic disorders. Early detection and understanding of these processes are essential for prevention and treatment. Understanding the cell cycle and its checkpoints help us comprehend how malfunctions lead to serious conditions like cancer and genetic disorders. While treatments and interventions are available, early detection remains our best defense against these conditions. Knowledge is power when it comes to health.
Thank you for joining this lesson on malfunctions during the cell cycle. Remember, knowledge is the first step toward prevention and understanding. If you found this video helpful, don't forget to share it with others who might benefit. Stay curious, keep learning, and see you in the next lesson. Thank you for watching!